Avian Influenza AI is a viral disease which represents one of the greatest concerns for public health in recent times. As well, the disease remains a major threat for the global poultry industry. Severe outbreaks of highly pathogenic avian influenza A H5N in birds throughout Asia, Africa, and Europe have resulted in human deaths, raising concern about its potential for adaptation to human as a result of extensive direct contact with infected birds. This situation has dramatically attracted the attention of the veterinary and medical scientific communities. Avian influenza is first of all a poultry disease affecting poultry production and international trade. The control of avian influenza really starts with control in poultry. Moreover, H5 viruses are evolving quickly. More than 10 genetic types or clades are already identified in different areas. Avian influenza control programs are based on biosecurity measures, stamping out policy or the implementation of vaccination programs. Vaccines available are inactivated produced from low pathogenic strains or through reverse genetics techniques. Recombinant vaccines using either smallpox virus or the Newcastle disease virus. However, despite its intensive use, the disease is still present in some countries because of the interference with maternally derived antibodies, MDA, which impairs very significantly the efficacy of any vaccine when applied at early age, the frequent antigenic changes of AI viruses, the uneven quality of vaccine application at the farms, the limited duration of immunity, making booster vaccinations needed. New solutions for improved and simplified control of avian influenza are needed. Ceva is a fast-growing animal health company based in Libourne, France. Ceva, through investments in research and development, owns a broad and unique poultry vaccine portfolio with conventional live and kill vaccines, immune complex and vector vaccines. Ceva Research has developed a new approach for avian influenza protection, Vectormune AI, using vector technology. A vector vaccine is a vaccine virus that is modified to express new antigens. Vectormune AI vector is a herpesvirus of Turkey, HVT, a well-known, safe and stable Marek vaccine. The donor virus is a H5N1 type. One of its genes, the amagglutinin HA gene, has been selected for its capacity to induce strong protection against avian influenza virus infection. The HA gene has been inserted inside the genome of the HVT virus without impairing the replication capability of the Marek's vaccine. Vectormune AI is a frozen cell-associated vaccine which needs to be stored in liquid nitrogen DIRA. Prior to use, the vaccine needs to be reconstituted with sterile diluent, a specific diluent for Marek's vaccines. The process begins with inhalation or ingestion of the AI virus. The virus particle binds to the host cell membrane by the amagglutinin HA protein. During the cell infection process, the virus particle is endocytosed. Then after chemical reaction, the HA protein is cleaved by cellular proteases. The HA protein causes the fusion of viral and cellular membranes, and the RNA of the virus is released inside the host cell. Thereby, the virus replicates inside the cell, which is then destroyed at the end of the replication cycle. The infected cells express the HA protein and multiple replication cycles occur with release of virus leading to the destruction of a large part of the infected tissue. As a result, these viruses can replicate in many organs of the bird, including the spleen, liver, lungs, kidneys, and brain. Vectormune AI has to be injected at day 1 subcutaneously in the back of the neck. The single vaccination stimulates simultaneously the immune response of the birds against avian influenza and Marek's disease. When a vaccinated bird comes into contact with an avian influenza virus, the infection is reduced by blocking the binding of the virus and the host cell membrane. Infected birds are then protected from avian influenza disease much better than by using conventional vaccines. Independent laboratory and field experiments have revealed that this vaccine can dramatically help improving the control of avian influenza. Vectormune AI has proven efficacy against 8 different clades of H5HPAI viruses from different geographic origins. Broad protection, demonstrated protection against different H5 avian influenza viruses isolates from several geographical origins and clades. Convenience, hatchery application at day 1 of Vectormune AI does not interfere with the maternally derived antibody status of the chicks. Long-lasting protection, the Varix virus HVT virus remains in the chick for life. Safety, the vaccine strain does not spread, shed or revert to virulence. The vaccine does not replicate in the respiratory tract. Use of an aqueous, no oily adjuvant.